Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This episode is going to cover the circulatory system and a little bit of fluids. So the first thing I'm going to talk about is anatomy, and I'll go over kind of the definition of veins and arteries and talk about blood flow and what that looks like throughout the body. Then I'll talk a lot about the heart. I'll cover a ton of different topics within the heart. And then lastly, I'll talk about how fluids, and I'm talking about physics fluids, can be applied to the circulatory system and what topics within fluids you're most likely to see. And I just want to mention too that I'm going to be talking about moving fluids, not fluids that are standing still. So maybe I'll do a podcast later and cover more broad topics from fluids, but this is only going to be about flowing fluids, not static fluids. So I'm not going to cover things like buoyancy, osmotic pressure, any of those things. And this podcast is a twofer. I'm going to cover two different MCAT sections within this podcast. That's going to be the chemistry physics section and also the biochem section. And I find this system, the circulatory system, to be probably the most interesting out of all the different systems. And I don't know why that is. Maybe it's the fact that there's so much physics involved in the heart and, pumping and pressure, and maybe that's why I like it. But anyways, this was one of the most fun episodes to write, and I hope that it helps you as you study, and I hope you find this stuff as interesting as I do. The circulatory system consists of blood vessels, and the heart. And its purpose is to deliver nutrients, oxygen, hormones to the cells in the body. In addition to this, it traffics waste products to different places in the body so that they can be excreted. And the circulatory system can be further divided up into two different circulatory systems. The first is systematic circulation, and this delivers blood to the organs and tissues in the body and mostly provides these cells with oxygens and other nutrients, as I was saying. And then you also have the pulmonary circulation, and that essentially carries blood to the lungs in order to be reoxygenated, so then it can be pumped back to the heart and then out into the systematic circulation so that it can provide these cells and tissues with oxygen. And so the first thing I want to get into here is just a little bit of anatomy and kind of go over some names and some structures and ordering within the circulatory system. So in terms of veins and arteries, you probably want to know the difference between these. You probably already do, but I'm going to reiterate that veins are blood vessels that carry blood to the heart, and arteries are blood vessels that carry blood away from the heart. And typically you like to think of veins as carrying deoxygenated blood and arteries as carrying oxygenated blood. However, there's an exception here, and that's either umbilical veins or pulmonary veins, and they carry oxygenated blood. And then pulmonary arteries and umbilical arteries carry deoxygenated blood. And why is this? That's just because they are carrying blood away from the heart. And so that's kind of the definition to remember. Just remember that veins carry blood to the heart, arteries carry blood away from the heart. And don't just solely base this off whether blood is oxygenated or deoxygenated. And so you can kind of think about it this way too. So the point of the pulmonary circulation is to reoxygenate the blood. So these vessels are going to go from the heart to the lungs to get reoxygenated. Well, what's going on? Well, blood is moving away from the heart. It is deoxygenated because it hasn't made it to the lungs yet. So therefore, because it's moving away from the heart, they are arteries. And then the same thing afterwards, right? This blood gets reoxygenated at the lungs, and then it shoots back up to the heart. And because this blood is going to the heart, it's traveling through veins, even though just this blood is oxygenated. And in terms of veins and arteries, there's a few different levels of organization depending upon how big these vessels are. But before I discuss each of these types of vessels, I just want to first talk about the path that blood takes as it travels from the heart through systematic circulation back to the heart. So it's first going to go through the aorta, and that is the biggest artery that's leaving the heart. From there, the aorta breaks down into different arteries, and then each artery breaks down into a bunch of different arterial, and then arterials branch out into capillaries, which then come back together to form venules, which then come together to form veins, which then veins come together to form the largest vein in the body, which is known as the vena cava. So I want to break each of these down for you. So starting with the aorta, as I said, that is the largest artery in the body, and that is directly leaving the left ventricle. And according to an online source, it would take about a minute for you to completely bleed out if you were to cut your aorta, which I don't know how that would happen, but would be devastating. The next level of organization is called an artery. So arteries are a bit smaller than the aorta, and they contain a layer of smooth muscle, which is very important for vasodilation and vasoconstriction. And I should mention too that the aorta does have smooth muscle. That is a feature of arteries and of arterials too. I'll get to that next. And this is in contrast to veins, which also do have smooth muscle, but the layer of smooth muscle within the veins is a lot smaller than the layer of smooth muscle in arteries. So keep that in mind. All right, and so the next layer of organization is called the arterial. It's a little bit smaller than an artery, and these have the greatest effect on overall blood pressure. And this is due to the fact that they are more rigid than a lot of the other bigger arteries in the body. And like other arteries, arterials have a layer of smooth muscle in the wall of the arterial. The next and smallest type of blood vessel is called a capillary. And capillaries are made up of a single layer of epithelial cells, which enables different molecules to diffuse into tissues. So, oxygen, glucose, water are able to kind of leak through this single layer of epithelial cell and make it into the different tissues that need these nutrients to be able to function properly. And then after the capillaries, you have what are known as venules. And venules are essentially just a very small type of vein. And as I said a little bit earlier, veins also contain a layer of smooth muscle within the wall of the vein, but it's a lot smaller of a layer compared to arteries. And then venules come together to form veins. And veins are larger than venules, have a lot of the same properties. As I said earlier, they carry blood back to the heart. And interestingly enough, veins and venules together contain 70% of the blood volume at any one time. So they act more as kind of like a blood storage system, or you can kind of think of them as capacitors a little bit if you're to make the comparison to circuits. And this is due to their capacity to be able to distend or to be able to expand and readily store high volumes of blood, even at low pressures. And then a bunch of veins come together to form the largest vein in the body, known as the vena cava. And more specifically, the largest vein in the body is the inferior vena cava, which carries deoxygenated blood from the lower half of the body back up to the heart. So let me summarize the levels of organization here in the path of blood as it makes its way from the heart through circulation back to the heart. So first it's going to go through the aorta, which is the largest artery. Then it's going to go into a bunch of smaller arteries, then into smaller arterioles, then into even smaller capillaries, and then into a little bit bigger of venules, then into bigger veins, and then into the biggest vein, which is the vena cava, and then back into the heart. The next thing within the circulatory system that deals with anatomy that I want to talk about is the anatomy of the heart, which I think is a bit more interesting than some of the other cardiovascular anatomy. So the heart is divided into right and left sides, and each right and left side has two different chambers, one of which is called the atrium, one of which is called the ventricle. And these two chambers are separated by valves. And these valves are designed to basically let blood pass through the valve one way, but then not flow back the opposite way through the valve. And as an engineer, I would call this type of valve a check valve. I don't know if anyone's heard that before, but that is what that is called in the engineering field. And the heart's four valves have different names. So there are two valves that are atrioventricular valves or AV valves. And then there are two valves that are called semilunar valves or SL valves. And these can be broken down further. The two AV valves are called the mitral valve or the bicuspid valve and the tricuspid valve. And then the two semilunar valves are further broken down into the aortic valve and the pulmonary valve. And I'll get into a little bit more of what exactly these valves do or where they are in the heart. But before I do that, you need to understand how blood flows through the heart. So let's start at the vena cava, which connects to the right atrium. And again, this is carrying deoxygenated blood back to the heart. So this deoxygenated blood passes from that vena cava into the right atrium. From there, it passes through the tricuspid valve and enters the right ventricle. Then from that point, this blood is pushed from the right ventricle through the pulmonary valve into the pulmonary arteries. And from there, it makes its way to the lungs where it gets reoxygenated. And then it goes back to the heart through the pulmonary veins and enters into the left atrium. Then it passes from the left atrium to the left ventricle through what is known as the bicuspid or mitral valve. And then last, once it's in that left ventricle, it gets pumped out into the systematic circulation through what is known as the aortic valve. And so that's the basic naming of these different valves. And I don't think that's super important for the MCAT, but you never know, you might see that come up. And those are pretty self-explanatory. I mean, you can think of the aortic valve being right before the aorta. There's a few that you might have to memorize, but really not that bad. What you should also understand is the general concept behind why the heart needs to have these one-way valves. And really it gets to the point that you need to be able to prevent backflow of blood in the opposite direction of circulation in the heart and in systematic circulation, right? Because if your blood is flowing the opposite direction that you want it to flow or that your body wants it to flow, it's not going to be a good thing. And so the way I like to think about it is think about this. So what is happening to the heart when it's pumping blood? Well, it is increasing in volume and therefore the pressure is very low when that's happening. It's like a vacuum. It's almost like zero pressure. And maybe it's negative pressure. It probably is negative pressure. I don't know. But what's happening is you're getting these strong decreases in pressure, and then all of a sudden it's contracting. That volume is getting smaller, and you're having a very large increase in pressure. So it's expansion, contraction, where your pressure in the heart is going up and then going way down, and then going up and then going way down. And so then what would happen if you didn't have this one-way valve? Let's say the aortic valve that separates the aorta from the left ventricle. As that pressure increases in the left ventricle, that's going to force blood out from the left ventricle into the aorta where it's going to go around circulation. And that pressure is actually going to be the driving force that pushes that blood around the body. And that's good. That's what you want. But then what happens after the heart contracts and then the volume of the heart expands, the pressure in the heart decreases? What's going to happen? You're going to lose the pressure gradient that exists between the left ventricle and the aorta. And now there is going to be a pressure gradient that reverses, right? The pressure is now going to be lower in the left ventricle, and blood from the aorta is going to want to then flow back up into the heart. And that's the opposite direction. You don't want that. And so what these one-way valves prevent is that exact situation. They basically close off the heart so that it can't have any backflow, and therefore prevents there from being this backwards pressure gradient that then draws blood up back into the heart after it's left the heart and gone into circulation. But it allows it to use that pressure gradient that's created when the heart contracts and produces high pressure in the heart to then be used to push the blood throughout the rest of the body. And for your interest, when blood is actually sucked back up into the heart, as I was describing, from the aorta, that is a condition that's known as aortic valve regurgitation. And I think really the takeaway here is to remember that pressure gradients determine the direction in which a fluid is to flow in pipes, in vessels, whether that's in the body or outside of the body. Fluids will flow from high pressure to low pressure. And so again, the heart is essentially this pump that's contracting, creating a high pressure situation, which then forces blood to go throughout the body because there's this pressure gradient. And so, in your veins, you have a much lower pressure than in your arteries. And so therefore blood is going to flow from the artery to the vein. And so you got to always think about these pressure gradients. And if I was to have a lower pressure in the heart than in the aorta, again, you're going to drive that regurgitation happening if you don't have this one-way check valve. So before I move on to talking about the heart and getting into a little bit more complex topics, I want to summarize the path of blood flow throughout the human body, taking into account the heart, taking into account all the different vessels. So deoxygenated blood enters the heart at the right atrium, then is pumped to the left ventricle, which is then pumped into the pulmonary arteries, which carries the blood to the lungs to then be reoxygenated, which then goes into the pulmonary veins, which then flows into the left atrium, which is then pumped into the left ventricle, which is then pumped into the aorta, which then goes to the arteries, which then goes to the arterials, which then goes to the capillaries, which then runs to the venules, then to the veins, then to the vena cava, then back to the right atrium. And the last thing I wanted to mention here are the heart sounds. So the heart has two different sounds. You may have heard of these. The first is the lub and the second is the dub. And that is also known as the first heart sound and the second heart sound. And so the closure of the AV valves is what causes the lub noise, whereas the closure of the SL or semilunar valves is what causes the dub or the second heart sound to occur. Lub dub, lub dub, lub dub, lub dub. So I already talked a bit about the anatomy of the heart. I want to get into the function of the heart. I want to talk about this Qrx complex that shows up on EKGs and EEGs. I want to talk about the cells that make up the heart, specifically what causes the heart to spontaneously beat. And this stuff is, to me at least, is really interesting. And then lastly, I want to talk about the regulation of blood pressure and the role that the heart plays in that regulation. And then also talk about some hormones that play a pretty big role in this regulation of blood pressure. And so I've kind of already talked about the function of the heart. It acts basically as a pump to move blood throughout the body. And you might be thinking to yourself, okay, but what exactly is the definition of a pump? And so by definition, pumps consume energy to perform mechanical work in order to raise the pressure within a fluid and therefore cause that fluid to move. And as I said a little bit earlier, the heart actually does this by squeezing and reducing the volume, which then produces a higher pressure within the ventricles that then drives the blood throughout the body. And again, what drives fluids to flow is differences in pressure, or pressure gradients. So for example, within your aorta, you have a mean pressure of about 100 millimeters of mercury, let's just say. Now, as we go down to your arterioles, that pressure is going to start to drop and you're going to have a pressure around 60 millimeters of mercury. And that is what is going to drive the blood from your aorta into your arterioles. And by the time we get to your capillaries, the pressure is about 20 millimeters of mercury. So it's even lower. And again, that is the driving force that drives the blood into the capillaries. And as you guessed, inside your veins, the pressure is even lower, somewhere in the 5 to 10 range of millimeters of mercury, which then keeps driving it until you get to your right atrium, which is going to be about 2 millimeters of mercury. So throughout your systematic circulation, then, you have a steadily decreasing pressure all the way back to your right atria, which drives the blood all the way around your body. So keep that in mind, that pressure gradients are this driving force. Don't forget that. If there's one takeaway from this podcast, remember that. And so this discussion on blood pressure brings me to two terms that you're going to want to know. So you may have noticed that I said your blood pressure in your aorta is somewhere about 100 millimeters of mercury. The mean pressure. And I got to say mean pressure because the pressure in your arteries is constantly shifting between two values as your heart beats and contracts and then relaxes. So when your heart is contracting, that is known as systolic pressure. And then when your heart is relaxing, that's known as diastolic pressure. And as I'm sure you could guess, systolic pressure tends to be a lot higher than diastolic pressure. And moreover, when you're given these blood pressure readings, say you're at your primary care provider or something like that, I'm sure you've heard them give you a blood pressure number of like 120/80, let's say. That's a pretty normal blood pressure. That is going to be the systolic pressure over the diastolic pressure. And this again is the pressure that is felt by the walls of the arteries. Because once you get to the arterioles, this pressure is already decreasing quite a bit. And also it starts to kind of dampen this two pressure thing you have going on in your arteries. The pressure is a little bit more narrow of a range between two values. And then by the time you have the capillaries, the pressure is just constant. There's no two different pressures depending upon whether your heart is contracting or relaxing. It's just a single constant pressure. And same goes for your veins. And I don't know why exactly that is, why this pressure difference kind of dampens over time. Might have to guess because maybe, the further out from the heart you get, that blood is not really feeling that contraction and relaxation as much as it is when it's near the heart, like in the aorta or in the left ventricle, but I don't really know. And before I move on to the heart's electrical system, I just want to mention the fact that giraffes actually have twice the blood pressure of humans, which is kind of crazy. I mean, I would think of us as having high blood pressure, I guess, and needless to say, giraffes have twice the blood pressure of humans, which is why they are all actually on aspirin regimens. All right, so let's briefly talk about the heart's electrical system. And what I want to talk about here is the path that this electrical signal takes throughout the heart. And when I talk about the different cells of the heart, I'll specifically talk about these pacemaker cells that create this signal that gets sent out and propagates throughout the heart. But just for this segment, just understand the path that this electrical signal takes. So the electrical signal itself is generated at the sinus node or the SA node. From there, it travels through the atria to the atrioventricular node, also called the AV node, and at this point, the atria contract. From there, it travels to the bundle of His, which branches off into left and right bundles as it travels down through the ventricles. And then near the bottom of the ventricles, this signal gets passed on to the Purkinje fibers, which causes the ventricles to contract from the bottom up, which is actually important for blood flow because it kind of directs the blood through these different valves out into circulation or out into the pulmonary circulation. And so to summarize that all up, what you need to know is that the signal within the heart that tells these cardiac cells in the heart to contract, it is generated at the SA node, which then travels to the AV node, then to the bundle of His, then to the Purkinje fibers. All right, so the next thing that I want to talk about in terms of the heart's electrical system is this Qrx complex that shows up on EKG waves. So these are these graphical blips or spikes that you may have seen, and they show up on these electrocardiograms, which are either EKGs or ECGs. Go look up a photo if you don't know what I'm talking about. But it's just this kind of spiky looking thing. Hard to describe. If it were to make a noise, it would probably be like bloop, 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 bloop. I hope that helps somehow. Great radio because, I'm sure you can picture exactly what I'm thinking. But so there's three different parts of the wave to know. That's going to be the P wave, the QRS complex, which is that big, tall spike part. And then you're going to want to know the T wave. So there's three different parts, and what you want to know is what is going on in the heart during these three parts. So during the P wave, which is this first short little wave in the EKG, what happens is the left and right atrium depolarize. And then the next segment of this wave, which is this QRS complex, represents ventricular depolarization and atrial repolarization. And this QRS complex really contains three different peaks. One of those is the Q wave, one of those is the R wave, and one of those is the S wave. Then lastly, we have the T wave, and the T wave represents ventricular repolarization. Another important thing to know is that the atria are in systole or are just starting to contract in the middle of the P wave. And this kind of corresponds to atrial polarization, right? Because polarization means that the cells are, or I guess the atria are contracting, right? The signal has reached the atria, and then these cells depolarize. The other thing to know is that at the R wave, which is this big spike, that is when you start ventricular systole, or the ventricles start to contract. And again, this makes sense, right? Because the Qrx complex represents ventricular depolarization. So therefore, contraction of the ventricles needs to start around that time. And now you know it starts at that R weight. All right, the next little bit that I want to talk about here is the types of cells that are in the heart. And there's two that I'm going to talk about. These are either cardiomyocytes or cardiac pacemaker cells. So those are the two I'm going to talk about. Not sure if those are the only two cell types in the heart. I'd assume no, but maybe everything does fall underneath those two categories. Don't know. Just know that those are the two types of cells I'm going to talk about right now. So cardiomyocytes are muscle cells of the heart, right? And you should recognize that little ending myocyte, which means muscle cell, and they make up the atria and the ventricles in the heart. And their one goal is to contract in unison so that the heart can pump blood effectively. And so how do they do that? How do they all know how to beat at the same time? Well, it all comes down to the fact that there are gap junctions between these muscle cells so that basically they can kind of share cytoplasms, right? These signals can kind of just pass between cells super easily through these gap junctions. And I was reading a research article about this, a review from the Cardiovascular Research Journal, volume 62, that said, quote, "A given cardiomyocyte in the adult working myocardium is electrically coupled to an average of about 11 adjacent cells, with gap junctions being predominantly localized at the intercalated discs at the ends of rod-shaped cells." It would go on to say that basically these gap junctions, in addition to the cellular architecture of cardiac tissue, which I'm guessing is like a uniform network of cardiomyocytes, are the two factors that really play a big role in the ability of cardiomyocytes to all contract at the same time. So again, the role of cardiomyocytes are just to really contract in unison and pump blood. Where shit gets really crazy is when we start talking about pacemaker cells. And these are really interesting. So these pacemaker cells reside within the SA node in the right atrium. So remember we talked about the SA node. That is where the signal is generated, the electrical signal is generated that spreads around the heart. And these cells are characterized by abundant expression of the hyperpolarization activated and cyclitic nucleotide gated ion channels, which is a damn mouthful. What that really means is that these pacemaker cells produce this very specific type of ion channel that is activated by hyperpolarization and performs in this kind of cyclical manner, right? Which is needed for a heartbeat, right? Because the heartbeat's very cyclical. It's just lub-dub, lub-dub, lub-dub. It's the same thing over and over and over and over. And these pacemaker cells undergo spontaneous depolarizations, which sounds essentially like magic, but this happens in a pretty well-defined manner, and this all has to do with voltage-gated ion channels, which were what we just talked about, which makes these cells unique. So essentially what happens is this, and I'm summarizing this, and I just want to say also that this is probably out of the scope of the MCAT. This is like really on the edge of what they could include. I really highly doubt it. But anyways, the first thing that happens in this process is that the sodium channels inside the membrane of these pacemaker cells opens. And at this point, sodium rushes into the cell, which starts to depolarize the cell, which means that the voltage is actually going to start increasing. Now, at some point, these sodium channels close and then calcium channels open. And this leads to a further depolarization of these pacemaker cells as the voltage gets more and more positive until eventually these calcium channels close, and then the potassium channel in these cells opens up and potassium rushes out of the cell. And so what happens there is then this cell is going to become repolarized, and it's going to go all the way back down to where it started out with in terms of its voltage, which is now going to be very negative relative to the outside of the cell. And at this point, the sodium channels open again, and the whole cycle just repeats itself. And this is what generates this rhythmic action potential that is sent out to all the cardiac myocytes that causes them to contract in this rhythmic fashion. And that's really a brief overview of how these pacemaker cells kind of work and generate these rhythmic signals. If you want to learn more, I'd go watch Khan Academy or something like this. I think this is a pretty low yield topic. So I'm going to move on now and talk about hormonal control of blood pressure and its relationship to the heart.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
So there are 5 different hormone systems I want to talk about. The first is the Renin-Angiotensin System. The second is Aldosterone System. The third is the Atrial Natriuretic Peptide System, then Vasopressin, and then the action of nitrous oxide. So let's first start by talking about the Renin-Angiotensin System. So I talked about this a bit in the kidney podcast, I think, the renal system podcast, I'm pretty sure. But this system is involved in the homeostasis of fluid volume and electrolyte composition of body fluids and helps control blood pressure. So renin is stored and synthesized in the kidney. More specifically, it is synthesized in the afferent arterial by juxtaglomerular cells. And these are specialized types of cells that have the capability of creating, storing, and secreting renin. And renin release is actually inhibited by stretching of the afferent arterial, what stretches the afferent arterial, which is going to be high blood pressure. So when you have high pressure in that part of the kidney, it's going to cause renin to be inhibited. On the other hand, renin secretion is going to be stimulated when there is a fall in blood pressure, and that's going to be again picked up by sensing pressure within the afferent arterial, or it's stimulated to be released when the body senses a decrease in sodium chloride within the blood. In addition to these two factors, renin can be stimulated to be released directly by nervous innervation or directly by nerves that stimulate this to happen. So, what exactly does renin do? Well, renin acts to convert angiotensinogen into angiotensin 1. Then, angiotensin converting enzyme converts the inactive angiotensin 1 into its active form, which is angiotensin 2. This active form of angiotensin binds to its receptor on endothelial cells and causes vasoconstriction, which is just narrowing of the blood vessels. And what does that do? Well, that increases blood pressure. So overall, renin is released when blood pressure is low, and that's sensed one of 3 different ways. And then it causes the blood vessels to narrow or contract, and that acts to then raise blood pressure. Aldosterone, on the other hand, is a steroid hormone that is secreted in the adrenal cortex of the adrenal gland, which sits right on top of the kidneys, and it acts primarily in the renal collecting ducts to stimulate the reabsorption of sodium as well as secretion of potassium and hydroxide ion. And if you think back to the hormone episode, or maybe in the kidney episode, I talked quite a bit about steroid hormones. And what these do is they bind intracellularly and affect gene expression. And just to give you an example of what exactly aldosterone does, here's a review paper by Dr. Muto of the Department of Nephrology at a Japanese medical school. It says, quote, transcription and subsequent translation result in the production of new proteins that modulate the activity of ionic transportation systems located in the apical and basolateral membranes of the target epithelial cells. In other words, when this hormone acts on the gene expression of cells within the kidneys, what it's going to do is it's going to cause the production of new proteins that go on and modulate the activity of ion transport systems, specifically of sodium ion transport systems, that then cause their intended effect. So as you know or are learning right now, reabsorption of sodium causes water to follow that sodium along with it and be reabsorbed. Water follows salt. You can kind of just remember that as a general rule within the kidneys. So what's going to happen then is the blood volume is going to increase because you're absorbing water back into the blood. So if you were to have, a set vessel, let's just say you have a square of metal and you just keep injecting more and more water in, what's going to happen? The pressure within that vessel is going to just keep increasing. So when your body reabsorbs this blood and the blood volume increases, you're going to raise the pressure in the blood. So you're going to raise the blood pressure. So to summarize that, aldosterone acts to increase blood pressure by increasing the reabsorption of sodium that then is followed by water. The next hormone that I want to talk about is called vasopressin, or antidiuretic hormone. And vasopressin, or antidiuretic hormone, is synthesized in the hypothalamus and is then secreted by the pituitary. And those are both in the brain. And this is a peptide hormone. And this is actually synthesized due to changes in blood osmolarity. So there's neurons in the hypothalamus that have osmoreceptors that can sense changes in blood osmolarity and then synthesize antidiuretic hormone in response to hyperosmolarity or high osmolarity. And if you think about that, that's just having too many salts or dissolved solids within the blood, that high osmolarity. So anyways, after the hypothalamus or neurons in the hypothalamus recognize this high blood osmolarity and release this vasopressin, what happens then is that this vasopressin makes its way down to the kidneys and binds to GPCRs, which are G protein coupled receptors in the distal tubule and the collecting duct. And remember, these are actually receptors on the surface of cells and cause aquaporins, which are water channels, to be inserted into the apical membrane. And this allows water to then be reabsorbed as it flows down its concentration gradient. Because remember, again, we're talking about when there is a lot of dissolved solids in the blood or there's a high osmolarity in the blood, right? Really concentrated blood, what's going to happen? Water is going to flow back in to the blood and just follow its concentration gradient. So what is this going to do? This is again going to increase the blood volume, which is going to increase blood pressure. And in addition to this action, it also acts directly on blood vessels, causing them to constrict, which again, this is called a vasoconstriction. And this is going to increase the blood pressure by increasing the total peripheral resistance. And I'm going to talk quite a bit more about total peripheral resistance when I get to the more physicsy part of this podcast, but just keep that in mind right now. When total peripheral resistance increases, so does blood pressure. The next and probably most interesting molecule that modulates blood pressure is what's called atrial natriuretic peptide, or ANP. And this is a small peptide, about 28 amino acids in length, that is secreted by the heart upon atrial stretch, which occurs when there is high systematic blood pressure. And what's so interesting about this molecule, at least to me, is that there are cells in the heart that can actually sense when the atria are becoming overstretched and are able to be like, oh, this isn't good. We have really high blood pressure right now. And then they're able to secrete this little protein that is going to be able to help reduce blood pressure. And so then what happens is that this little ANP protein goes out and it causes vasodilation or expansion of blood vessels in the body. And it also increases glomerular filtration rate, which basically helps the body excrete more fluid from the blood and therefore decrease the fluid volume. And then it also increases the renal excretion of both sodium and water. And again, this is to decrease the fluid volume in the blood. And so overall, what this ANP protein is going to do is it's going to lower total peripheral resistance via vasodilation, and it's going to decrease blood volume. And both of those are going to act to actually decrease the blood pressure in the body. All right, and the very last molecule I want to talk about here is called nitric oxide, or NO. If you're anything like me, or as much of a douchebag as me, then you will know Nitrous oxide is typically an ingredient in pre-workout. It gives you a sick pump because it is a vasodilator. And basically, in short, nitric oxide is a mediator of vasodilation in blood vessels. And this pathway is pretty complex. I was reading on it a little bit, and I just decided to say, you know what? Just know that nitric oxide is a vasodilator and contributes to the lowering of blood pressure in the body. And interestingly, I was reading a paper about the oral nitrite pathway, and I hadn't heard of this before, and it's kind of interesting. So about 25% of the circulating nitrite in the human body is being actively absorbed in the salivary glands from different species of oral bacteria that actually reduce nitrate to nitrite. And then we swallow the nitrite, which is then taken up in our GI tract, and then in the blood, nitrite can be reduced to nitric oxide by several different enzymes, and this can lead to vasodilation. And so what they wanted to know was why you get this phenomena known as post-exercise hypotension after you work out. And basically all that is that after you work out, you get this lowering of blood pressure for a few hours after the workout, and then you go back to a normal blood pressure. They wanted to know why that was. And they hypothesized that that was due to nitric oxide that was being produced by the oral bacteria. And so they wanted to test this in people, though. And so how would you kill all the bacteria in the mouths of someone and compare that to someone who didn't have the oral bacteria killed in their mouths after a workout, and just see if that has any effect? How would you do that? Well, you would use mouthwash, right? Mouthwash are going to kill a lot of the bacteria. So what they had people do is they had people work out, and this was running at a moderate intensity on a treadmill. And then they had half these people basically take mouthwash after, kill the bacteria in their mouth, and half of these people not kill the bacteria in their mouth. And they saw, and I should mention too, they used a placebo mouthwash. I don't know what that is. I'm guessing something that kind of stings a little bit so you don't know if you're taking a mouthwash or not. But after they had done this, they did see that the group that did not do the mouthwash had this lowering in blood pressure, which is what they expected. That's good. And this lowering was about 3 millimeters of mercury over a period of about 2 or 3 hours. What they did notice, and this is the interesting part too, is that the people that did the mouthwash had no lowering of blood pressure. So they went on to say that, well, these bacteria in the mouth are contributing to the lowering of blood pressure after you work out. And basically this is happening by oral bacteria that are secreting precursors to nitric oxide, which then get converted to nitric oxide in the blood. So pretty interesting story. Really, the takeaway message is that if you want the beneficial effects of this post-exercise hypotension, don't use mouthwash after you work out, because you're just going to be killing bacteria that can provide these important precursors to nitric oxide. So the last thing I want to do in this podcast is I want to apply some of the fluids topics, physics fluids topics, to the cardiovascular system. So I had to take a whole class on fluids during undergrad, and the professor's name that I had was Dr. Heinz, and he had this really thick German accent, and he spoke super soft, and he would like laugh at jokes he made pretty much constantly during class. And so he was super hard to understand, and it was just a horrible experience. And so therefore I kind of hated fluids, but I think it's kind of interesting when you can apply them to the human body and start to think about these things in terms of the human body. So hopefully this will be at least a little bit interesting to you. And I don't have a thick German accent, so you should be good. I'm going to talk about a few different topics. I'll start with total peripheral resistance. I'll talk a bit about viscosity. I'll talk about the continuity equation. And then I'll talk a little bit about the Bernoulli equation. And then before that, I'll just kind of define a few terms that you might see on the MCAT. The first thing I want to talk about is what an ideal fluid is versus a real fluid. So an ideal fluid is a fluid that possesses the following properties. It is incompressible, has zero viscosity, and no surface tension. And it's also completely made up, right? There is no such thing in real life as an ideal fluid, right? Every fluid is going to be a little bit compressible and is definitely going to have some kind of viscosity, especially blood, right? Blood is pretty viscous. So keep that in mind. Some equations only apply to ideal fluids, whereas some equations apply to both ideal and real fluids. Two other terms related to fluids that you might see come up are laminar flow and turbulent flow. And these describe the flow regime of a fluid. And so laminar flow refers to orderly flow in which there is little to no mixing between adjacent fluid layers. So you could imagine taking a drop of food coloring and dropping it into fluid that's flowing through a pipe. If this is laminar flow, you are going to have basically a completely straight line, right? There's going to be no mixing up and down in the flow regime in that pipe. However, if you have turbulent flow, this is a more random flow in which you do have mixing between adjacent fluid layers. And if you were to take a drop of food coloring and drop it into a pipe in which there is turbulent flow, you would see this food coloring kind of spread out and go in a bunch of random directions instead of just going straight, completely, perfectly linear with flow. Another way to imagine laminar versus turbulent flow is you can imagine laminar being like barely turning on the kitchen sink. You get this nice, pretty looking flow of water versus spraying water out of a hose, right? It's just going in random directions. And just so you know, blood that is flowing through blood vessels in the body is undergoing the laminar flow regime. The next topic that I want to talk about is total peripheral resistance, which should remind you of a topic within circuits, and that is resistors. And so resistors are circuit elements that resist current. So they tend to decrease current. And so just like a resistor, total peripheral resistance refers to the resistance to blood flow that is offered by all of the systematic vasculature. That includes veins, arteries, and capillaries, but excludes the pulmonary vasculature. And so you can imagine blood running through some vessel, let's say an artery. Well, as the blood contacts the walls of that artery, the walls of that artery offer resistance to it, right? It kind of pulls back on the blood as it flows. If this was to be an ideal situation, you can imagine like there's no friction between the walls of the arteries and of the blood, then the blood flow would not feel any resistance. But because there's interaction between the walls of the artery and the blood, it kind of pulls back on the blood and offers some resistance to that blood as it travels through these vessels. And again, the total peripheral resistance refers to the resistance offered by every single artery, vein, and capillary, excluding the pulmonary system, in the whole body. So it's this big total resistance overall, this big number from all the millions, billions, however many of these vessels there are in the body. And there's an equation to know if we're talking about resistance, specifically total peripheral resistance within the body. And this equation is called Poises Equation. And if you're looking at this and trying to sound that out, it looks like it's spelled Poisuli. So, and this equation says that the flow rate of blood through a vessel is equal to pi times the pressure times the radius of that vessel to the fourth power divided by 8 times the fluid viscosity times the length of that vessel. And while you would be given that equation, what's important to know is the basic proportionalities of the equation, and specifically of resistance. So I'm going to go through right now how resistance changes based upon these different variables. So the resistance within the vessel is proportional to the viscosity times the length of the vessel over the radius to the fourth. That is what the resistance is proportional to. Meaning that if we increase the viscosity of the blood that's running through these vessels, we're going to increase the resistance. If we increase the length of the vessels, we're going to increase resistance. If we decrease the radius of the vessel, i.e. we have some kind of vasoconstriction, we're going to increase resistance by a power of 4, which is a lot. And that to me is the biggest takeaway of this equation, is that if we increase or decrease the radius of the vessel, we are changing the resistance by that power of 4. So that's increasing by a lot, meaning that if we just double the radius, we are decreasing the resistance by 32 times. That's a ton. So really, if you want to change the total peripheral resistance, the easiest way to do that is to either make the vessels bigger or smaller in terms of their radius. And how do we do that? That's just vasodilation or vasoconstriction. It's also important to note that total peripheral resistance can be calculated given mean arterial pressure and cardiac output. And so cardiac output is equal to heart rate times stroke volume. And then obviously mean arterial pressure is just going to be the mean between your systolic pressure and your diastolic pressure. And so you can see there, then if I rearrange that equation, we have that the total peripheral resistance is equal to mean arterial pressure times heart rate times stroke volume. So what does that mean? Well, that means that if we have a higher blood pressure, we're going to have more resistance. Okay, that makes sense. That means if our heart rate increases, we're going to have higher total peripheral resistance. Makes sense. And then if our stroke volume increases, we're also going to have a higher total peripheral resistance. And to me, I think this is because you're going to have more blood going through. Your velocity of blood going through is going to be higher. And when you have higher velocity, you're going to have more of this resistance to flow. So really the 4 key takeaways for total peripheral resistance are as follows. Vasoconstriction increases total peripheral resistance. Vasodilation reduces total peripheral resistance. 3 is that the longer the vessel system, i.e. the taller the person, the higher the total peripheral resistance. And then the 4th takeaway is that the faster the heartbeat, the higher the total peripheral resistance. All right, the next concept I want to talk about here is viscosity. So viscosity is a fluid's resistance to flow. Think about it this way. If I take honey and I put it in a pipe, it would take a whole lot of energy in order to make that flow, right? And honey's got high viscosity. Now, if I take water and I put that into the same pipe and now try to pump that water, it's going to take a lot less energy. And that's because that water is less resistive to flow. I don't have to put in as much energy. It's got lower viscosity. And in terms of viscosity's physicsy definition, it's this complicated equation that basically says that viscosity is shear stress divided by the velocity gradient. You don't really need to know that. Something that does come up sometimes is the definition of a Newtonian fluid and a non-Newtonian fluid, and this does have to do with viscosity. So a Newtonian fluid is a fluid whose viscosity is constant no matter how much stress you put on it. In other words, the shear rate is directly proportional to the shear stress. And that's honestly a little bit hard to imagine. I'm sure you're thinking, okay, but what does that really mean? Well, I'll give you an example that water is a Newtonian fluid. The viscosity is constant no matter the stress you put on it. It's easier to see when you think about non-Newtonian fluids. So non-Newtonian fluids are fluids whose viscosity changes with the force that is applied to it. In other words, its shear rate is not proportional to its shear stress. An example here is that stuff that's known as oobleck. And I don't know if you've made this recently, maybe in like 5th grade you made it, but it's the substance that is made out of like cornstarch and soap or something, I forget. But what happens is if you really punch it really hard, like put a lot of force on it, it is really solid. Like you could maybe break your hand by trying to punch it. But if you just take your finger and just kind of like slowly let it go in the oobleck, it easily goes in. It's like really liquidy. So its viscosity changes depending upon the force that's applied to it. So if it helps you to remember this oobleck example of this weird liquidy solid stuff, remember that. You can also remember that blood is technically a non-Newtonian fluid as well. It's also important to note that viscosity decreases with increasing temperature. So if for some reason you want to decrease the viscosity in your blood because maybe your heart's having troubles pumping it super fast, one way to do that would be to raise your body temperature. Now, I'm sure that probably wouldn't be good for you to raise your body temperature up to like 105 degrees or something like that. But what that would do is that would decrease the viscosity of your blood and make your blood flow a bit easier. Interestingly enough, aspirin is a medication that actually lowers blood viscosity, which is why it lowers blood pressure. I mean, it can be used to sometimes prevent heart attacks and things like that. It requires less force from your heart to pump that blood around, which obviously is good if you have a weak heart that is, about to go into a heart attack. All right, the next thing I want to talk about here is what's known as the continuity equation. And this is a really big one for the MCAT. I've definitely seen this come up a few times. And really, this equation is just a mass balance. It just says the rate at which mass enters the system must be equal to the rate at which mass leaves this system. And this equation is expressed as A1V1 is equal to A2V2. And it just says that at some point 1, where the cross-sectional area is A1 and the velocity is V1, the product of that must be equal to the area or cross-sectional area times the velocity at some other point 2. And that is within a closed pipe system or closed vessel system. Conceptually, what this is saying is that as the cross-sectional area of a blood vessel changes, then so must the velocity of the blood that's traveling through it. So let's say, for instance, that blood is traveling in an artery when it comes upon this blockage in the artery. And at this point, the cross-section of the artery gets a lot thinner, and there's this only teeny little place where the blood can kind of squirt through into the opposite side of that blockage. And so what happens at this point, or so says the continuity equation, is that blood must speed up as it goes through that little opening in the artery. And why is that? Well, that's because the cross-sectional area decreases. Well, then the velocity must increase. Another interesting place this shows up is when we try to compare the blood velocity in arteries versus capillaries versus veins. If you could guess based on the radius of these different vessels, you would guess that capillaries have the fastest blood flow, right? Because they have the smallest radius. You would be dead wrong, actually. Capillaries have the slowest blood velocity because we have a massive amount of capillaries, so the total cross-sectional volume of all the capillaries is larger than the total cross-sectional area of all the arteries, which is why blood flow is slower. And it's a good thing that blood flow is slower because that gives us more time to exchange nutrients between the blood and the different tissues that capillaries run through. It's also kind of weird to think about that the blood velocity increases in the veins after it's been through the capillaries. So what happens is that you essentially have blood flowing fast through the arteries and then slows down in the capillaries, then speeds back up in the veins. It's kind of weird to think about, right? I haven't ever thought about that. So in terms of the continuity equation, be able to know how to apply this A1V1 equal to A2V2. It's a pretty simple equation. I think I actually used that on the MCAT, like I had to do a real calculation, and be able to know what that means in terms of if I increase the area, what's going to happen to the velocity. The last and final concept I want to introduce here, equation I want to introduce, is the Bernoulli equation. And the Bernoulli equation is essentially an energy balance. And it just says that the total energy within a closed fluid flow system must remain the same. And this only applies to ideal fluids. And again, you would be given this equation on the MCAT, so I would not spend your time trying to memorize the Bernoulli equation and understand how to apply it and stuff. I really don't think you'd see it for calculations, and if you did, it'd be a pretty easy plug and chug problem. And essentially what the Bernoulli equation says is that the pressure energy in the system plus the kinetic energy of the flowing fluid plus the potential energy of the flowing fluid must remain the same over time, no matter what, right? Because energy can neither be created nor destroyed, it's conserved. And in terms of variables, it says that pressure plus 1/2 times the density times the velocity squared of the fluid plus the density times the gravitational constant times the height of the fluid must be equal to some constant. And again, all this math stuff really doesn't mean too much for the MCAT because you'll be given this equation and really it's going to be like plug in density, calculate the height of the fluid you're going to need to produce some given velocity. It's not going to be that involved. You don't really need to know this equation. One thing that the Bernoulli equation does show, and one takeaway that I would have from this, is that the Bernoulli equation shows that when a fluid speeds up, the local pressure of that fluid drops. So think about the pressure at a blood vessel blockage, kind of what I was just talking about. The pressure at that blockage would actually be lower than the pressure in the vessel surrounding where there wasn't a blockage. And that's because this fluid speeds up through that blockage. And that's kind of counterintuitive, right? You'd think there'd be a really high pressure because you have a blockage, right, at that blockage. But no, there's actually a really low local pressure at that blockage. So that's something that's kind of counterintuitive and that I would take away from this. Other than that, just be aware of this equation. It's an energy balance. You might need to use it to do different calculations. All it says is that the energy within a flowing fluid is constant. All right, and with that, I conclude the Circulatory and Fluids podcast. One thing you'll notice I didn't go through was some of the statics fluid topics, like osmolarity, buoyancy, some of those other important things. So I might do a podcast later going through some of those other fluids topics and also bringing up maybe some of these ones in greater detail because I kind of just really introduced them and showed some of their applications to the circulatory system. Thanks for listening to the podcast as always. If you found this helpful, go tell a friend, go leave us a review on iTunes. All that stuff really helps. And, it's always good to see emails from people if you really feel like this helped you. Stay tuned for the next one. Should have an interview coming out fairly soon, I believe, hopefully. Thanks again for listening. See you next time.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on